זהו, זה מסיים?
זה מופיע.
בסדר.
זה מצלם.
מצלם. דריה. לא, זה. כן, כן, כן. בסדר. בסדר. גם אני מצלמת. אבל למה כבר זה התחיל? כי אני ביקשתי להתחיל. שירות המתארגנים. אני אזוז. חמוד. הנה אבי. כן. אני אפסיק ואחר כך אני אצא עוד פעם. איך עושים? איך מפסיקים? לא יודעת? אז אני לא מפסיקה. אם אני אפסיק, אחר כך זה לא יהיה להם.
זה נראה לי זמן סתם וזה כבר עובר.
בסדר, שיהנו מתארגנים. אוי, הם עושים לייקים, מה זה? הם עושים לייקים.
כן, הם עושים לכם לבבות.
שומעים אותי כשאני... ברור. נהדר.
בלייב.
כן, פעם ראשונה בחיים. ההתרגשות פה בשמיים והלייקים שלכם והלבבות שלכם כיפים גדולים. יש פה התרגשות מטורפת, מטורפת.
Sorry.
Sorry, it's okay. עם הטרנינג האדום זה השופטים, ומתיידדים עכשיו עם השופטת. יש פה בצד עוד הרבה שופטים. רגע, אני אראה לכם. הנה, שם בפינה. הנה, הם עושים לכם שלום. איזה התרגשות. I'm going over. כתוב לי.
תותחים, פשוט.
אין. ורד כותבת ששומעים אותי, היא לא יודעת. ורד יקירתי, שזו פעם ראשונה בחיים שלי שאני מתעסקת בפייסבוק.
אז כשהם מתחילים אנחנו נשתוק, נכון יפה?
ברור.
זה די והבן של יפה. בהצלחה.
ובהתרגשות כזו הכל מותר.
נכון. והילדים מוכנים.
כן,
שיהיה להם המון הצלחה.
בהצלחה.
מתים
מתוקים.
So I'm going to tell about myself.
חי נכנסו לאולם והם התקבלו בשמחה על ידי הילדים שלנו.
Okay, well, welcome. Well, one of the big advantages of iGEM is having high school young people here who are the forefront of innovation. So our first presenter today, Danci-K8 from Israel.
Good morning, I'm Itai from Danci-K8 High School, Israel. We are going to tell you what we did last summer. I might only be 16 years old, but I don't understand what Mr. Maslow formulated decades ago. I know the existence of mankind relies on a number of existence needs. The girls in our class are pretty busy with extra eating, how to minimize eating, while the boys are not bothered by it at all. They are troubled by another essential need. Today, we have chosen to focus on the need of being, nutrition. Obesity, diabetes, tooth decay, and intestinal diseases are excellent examples of common diseases caused by nutritional habits. Our project Flavors had two challenges. One, to develop an assay which can determine the taste sense receptors using a variety of alleles. Two, test the possibility to modify the desired alleles as a feasibility test to change people's taste sensing. Flavors will open new horizons in the study of nutrition, innovative assistance in the field of nutrition, which prevents and can even help people who cannot swallow pills due to its bitter taste. So, let's spray the taste away.
Thank you, Itai. I'm Mine. Humans are able to distinguish between several types of tastes. The tongue is composed from a top layer of many varied taste buds. The taste receptor cells, TRCs, are found on the surface of the tongue and the gums. The TRCs, together with the gustatory cells, send signals to the brain via nerve tips. The brain decodes the signals and creates the flavor image. The ability of humans to sense food taste is driven in part by genetic polymorphism in the taste receptor cells, organized in taste buds located within gustatory papillae. Genetic research will allow distinguishing a variety of alleles that determine the taste of food. For example, I like sweet, so I can assume that I have a high number of the TRCs that recognize this taste. Now my friend Avital will carry on.
Thank you, Man.
The work in the lab was divided into two parts. Stage 1 includes a unique characterization of the composition of test receptors alleles for 5 different tests. Stage 2, development of genetic analysis of the ability to test food ingredients, followed by in vitro editing of test-related SNPs by CRISPR. A. We perform the saliva sample into a unique membrane. Each of the 73 participants answered the questionnaire, which included experience for the 5 tests. B. Designing 15 SNPs. In order to characterize the collected DNA sequence in relation to the different tests, we decided to get help with SNP sequence. And C. Execution of Tuckman assay. Single nucleotide polymorphism in genes encoding to test receptors have been found to influence people's abilities to test flavors, coding SNPs that give rise to various haplotypes in these genes. Distinct phenotypes are associated with specific haplotypes, demonstrating that these SNPs have direct influence on test sensitivity or insensitivity. These haplotypes can explain the distribution of flavor preferences. To the best of our knowledge, this is the first research that examines a variety of SNPs simultaneously. Each of the previous studies examined the genetic dispersion of a particular test. This statistic made it possible to use modern technology called Tuckman. You can see that the same test has more than one SNP respectively to the specified gene. On the right, the negative results of the Tuckman method are without a mark. This is because the mark probe did not find a match to the sequence. On the left, the positive results where a mark was created due to a match between mark and DNA sequence. By using this method, we checked the presence of the different test alleles using the 15 pre-designed SNPs. The test was performed on 73 DNA samples collected. The goal of the test was to develop a unique panel to characterize the sense of taste. This panel has 2 parts. Each point from one part is connected to every point in the second part. This way we can create a meeting between SNP and DNA fragments examined. One of the aims of our project is to create a prototype panel that can characterize the composition in the sense of test receptors for each patient. And now my teammate Tomer will continue.
Thank you, Avital. The second stage of the lab included the identification of the selected allele for a specific test receptor encoded and sliced in vitro using the Cas9 RNA-guided enzyme. This change will allow us to control and navigate the ability of the sense of taste as needed. 5 synthetic gRNA molecules. These molecules were specially designed so that 4 of them were able to target SNPs in 4 flavors: grapefruit, coffee bitterness, NaCl and sour, umami and monosodium glucan respectively. And the 5th molecules will be used as a negative control. Negative control was from the human's gene scenic, and there is no high homology between that sequence and any of the SNPs. The CRISPR method. The simplicity of the CRISPR nucleus with only 3 needed components, Cas9 along with the crRNA and trace RNA, makes the system amenable to adaptation for genome editing. Pac57 plasmid. We designed 2 synthetic DNA constructs. The 2 constructs were cloned separately into 2 Pac57 plasmid. The first construct contained 15 wild-type SNPs called TASTE. The second contained 15 mutant SNPs called non-TASTE that we used as a negative control. So, let's sum up. Our experimental system consisted of 2 parts. First part, sampling 73 participants, ordering 15 SNPs, and performing Tuckman reaction in order to characterize the test sense system using a unique panel. The second part included testing the CRISPR system created in order to detect specific alleles. included testing a CRISPR system created to detect specific alleles For that, we engineered 5 gRNA, PAC57 plasmid to monitor positivity and negativity and performing CRISPR reaction. And now my teammate Gal will continue with the result.
Thank you, Tomer. Using the TAC method, we examined the polymorphisms of 15 alleles that characterize specific receptors to 8 tested flavors. The samples were tested in the lab to understand the distribution of each allele in the population studied. Each allele characterized whether it was heterozygote or homozygote. The unique part of this section was assembling a unique panel that characterizes a number of receptors to identify the taste with predefined alleles. In the current experimental group, no polymorphism was found for the 2 alleles for the umami taste in the group of the population tested. In checking the bitterness of coffee, we found that the 5 alleles checked in this test are heterozygote. You can see this in the graph shown here, received at the end of the process. Here, for example, the presence of the 3 colors indicates the presence of a variety of alleles, and so it will be defined as the tested alleles. Now my teammate Maya will continue with the results.
Sensitivity to the bitter taste is composed as PTC. This character is essential in avoiding eating poisons like in prehistoric times. People with higher taste sensitivity to bitterness are unable to swallow pills. We can see the distribution of the 3 alleles of PTC. It turns out the dispersion of these alleles are relatively similar in relation to the population. The distribution of 4 interesting tastes are shown in the next graph. We can see that close to 60% of the participants are not sensitive to alcohol. However, 8% of the participants are sensitive to the taste of alcohol. Alleles for the sweet taste are distributed the same in population with sensitivity and without sensitivity to the sweet taste. 5% of the population tested cannot tolerate the taste of coriander, which for them tastes like soap. 31% of the participants are sensitive to the taste of grapefruit, while 19.5% are insensitive to the taste. Now Tomer will continue with the electrophoresis results.
Thank you, Mai. The electrophoresis result showed the ability of the CRISPR system we built to perform 2 actions. First action, to identify the unique allele inserted into plasmid PAC57. The second action, to split the allele using Cas9. The electrophoresis shown present the 4 tested alleles. All of them split clearly by Cas9. However, the original plasmid was not split and remained whole. We sent and documented 38 biobricks to the registry. We added 3 functional collections of biobricks. 1. CRISPR-Cas9 collection. 2. TACMan assay of the FluidGEM nanofluidic gene dynamic chip collection. 3. Control for the TACMan assay and performing CRISPR on non-taster SNPs collection. And now I would like to invite Shachar to the stage.
Thank you. Now let's talk about the achievement. 1. This is the first research of its kind which analyzes simultaneously polymorphism of a variety of alleles of different tastes. 2. We find a significant match between the genetic polymorphism of the allele to the phenotype of the participant, and therefore the use of this unique panel will assist humans in several ways in the future. 3. It will be possible to use this method to develop a new area defined as preventive nutrition. The Flavov team offers a new system for proper nutrition, which will include, in the first stage, identifying existing alleles and then shutting off certain tastes as needed using the CRISPR system. The diet of tomorrow will allow us to spread the sense of taste and change it as we want. Now my friend Shir is going to carry on. Thanks, Shachar. Israel is an immigrant country. The citizens come from all over the globe, west and east. Some of them live in mixed settlements, and some still maintain a conservative cultural character and therefore genetic as well. Since the sense of taste has a genetic basis, depending on origin, we chose to do a number of social activities and a few regular sentences. The settlements we chose reflect the degree of statistical dispersion, which represent the general population in terms of affinity for taste, type of settlements, different ethnicity. We chose different age group for comparison, as well as the blind population, where the sense of taste should be sharper. Children. Studies prove that the number of taste buds in children is twice as more than in adults. Different ethnic origin. Different populations around the world are associated with a different affinity to various flavors and spices. The elderly. Over the years, this has changed and become dull. The blind. Represent a population group where the sense of taste should be stronger due to affected eyesight. At every one of the places, we conducted activities related to the sense of taste and, of course, give a broad and detailed explanation of the essence of synthetic biology. All the elderly, without exception, admitted to a change in their affinity to different tastes over the years. The age of the elderly was from 80 to 97. The blind. People between the age 22 to 45 come to the center. We visited the blind center in order to examine their sharpness in the taste sense in comparison to people who can see. Masada village. Masada village is on the Golan Heights bordering Israel and Syria. The village is a Druze village that is part of close society for thousands of years. Their common origin is Syria. We assume that on a survey level, we found differences between children from Masada and other Israeli children, and this is the reason we visited Kibbutz HaGoshrim and the elementary school in the city of Kiryat Shmona. Now my friend Daniel will close up.
Thank you, Shir. Okay. We collaborated with a few teams. We collaborated with a few iGEM teams this year. We participate at the mini jamboree at the iGEM with the Israeli teams. We collaborated with Team Mingado. We filled out survey together, and we mentored the Tel Hai College from the start. In order to reach the awareness of more and more people, our scientific concept and practice of gene therapy was also spread by the media, radio, social media sites, newspaper, TV, and meeting with school and the general public. We have about 1,400 students in our school. 1/3 of them are in classes of excellence. For the trip, we gathered the brightest pupils in science and math studies in order to present our scientific work, at the same time to clarify the position regarding the ethical question. Is gene therapy worthwhile only to save a life, or in cases of improving quality of life as well? The financial difficulty, the security, the closest to ISIS based in Syria, the physical distance create a mental distance from the option to achieve goals. And actually, a large number of our friends find themselves in the endless cycle of life where there is no line of progress. The ability to access information, to make it possible to participate in innovative activities. But costly, like the iGEM competition, is not obvious. We operated and explained in detail to empower the local resident and to prove, especially for the young people, is the sky the limit? We are here, aren't we? So the sky is not the limit. Flav off may change the face of the future nutrition. This is an exceptional combination of genetics and diet, and is actually a transition to a new world, a smarter world, a more nutritious and a modern world. We would like to thank Professor Daniel Berkovich, GGA Labs manager, the generous donor who enabled us to realize our dream to be here today. And lastly, the school staff who didn't leave us for a minute, even through the summer vacation. A special thanks to iGEM for holding this great event for all of us. Thank you for listening.
עכשיו אנחנו עוברים לחלק של השאלות.
Hello.
הם היו מדהימים.
Did you find any association between your results about haplotypes and characteristics of the people you study about maybe any disease or obesity or something like that? Is there any association between SNPs and obesity or any disease in the people that you've studied?
SNPs and obesity. Someone want to answer?
It's a very good question, but we didn't check it. But we will think about it.
I was wondering if you could share with us a little more detail on how your Cas9 complexes were generated and utilized.
We had the guide RNA that actually told the Cas9 system where to cut the SNP, and we had the found sequence to also specifically tell the right nucleotide to cut. And this is how it was very specific, and this is how the system actually works. We have the Cas9 enzyme, we have the SNP, we have the guide RNA, and that's it. This is what it's pretty simple.
Just to clarify, where did you get your Cas9 enzyme?
We ordered it as an active enzyme.
Oh, okay.
So I believe that taste can be also like your body's defense mechanism from allergens and spoiled food and everything. Have you guys thought about any side effects if your method work? Not only taste food, it could be your body's defense mechanism from food that's not good anymore, or like allergens you might have. You guys think of any side effects that if your method works and what actually.
During of the process, we didn't thought about this side, but I think we'll take it into consideration, and we will think about it.
Have you thought about it's an ethical question, so this is a big question, about using CRISPR on people? Because that's what you're ultimately proposing, right? So you're introducing the ability to edit our genome consciously. Have you thought about what that means to society?
Yeah, we actually held a conference at our school and we showed the negativity and positivity sides of the ethical question, and we did thought about it, but I think it will the advantages overcome the disadvantages, and I think it will help a lot of people.
I have a question. At DARPA, we did a study on congenital blind babies and taste, and we found that taste, the map of the tongue, is actually linked to touch. Maybe something for you to consider if you haven't already.
Yeah, we didn't think about it.
So the baby can visualize
Yeah.
the mother from the tongue when they're blind.
It's pretty smart.
One question time from the audience?
Yeah.
Yes, sir.
What is the application of the product to the general public?
Well, in the future, we're going to have a spray that you can spray the taste, your tongue actually, and it will change the taste and shut off certain taste as needed. This is what we vision. Thank you.
Thank you so much.
טוב, ברשותכם אני מכבה את הלייב. Davey, מכבה. Thank you very much.